live stream. Okay, it's done.
We have a lot of people still joining.
Mm-hmm.
Well, I see that our three welcome speakers.
Are we?
Yeah.
They are here. Okay.
Yeah.
Yeah, we have over 110 individuals. They are still joining. It's ticking up as I speak. I don't know how long we want to wait or if we want to start.
I think we probably want to go ahead and start. And so this is Emily Devereaux. I'm Executive Director of Research and Technology Transfer for Arkansas State University. And I'm one of the administrators for the Center for No Boundary Thinking. And we would like to welcome you today to the Arkansas Artificial Intelligence Campus Phase 1 Demonstration Showcase for Fall 2020. And thank you for joining us today. And I'd like to turn it over to Dr. Kelly Damphus, the Chancellor at Arkansas State University, for a welcome and short address.
Thank you very much, Emily. It's so great to see everyone here. I would think that this may be one of the bigger opportunities for the AI campus to gather. And I think sometimes I'm always looking for silver linings with COVID-19. And what I'm discovering is our ability to Zoom into conferences like this is actually increasing participation. I know that I teach a class every week and I have a in-person class and the attendance is pretty good in there. But I also do Zooms for people who can't come to class, and the attendance there is always 100% strong because you know, it's easier for them to get there. If they sleep in, they can certainly wake up and get on there pretty quickly. And so it's great to see a lot of people here today. It's my honor to welcome you here to this conference, and I really think that the AI Campus Initiative is a really unique piece of the Arkansas Research portfolio. I see Brian Barnhouse is here from the Arkansas Research Alliance, and their effort there is to bring researchers from across the state together to better understand how we can, instead of being competitive with each other, be actually complementary in our work. And I think the AI Campus Initiative is just one example of how this works. We have 6 institutions that are represented here today that will be presenting, and we've got participants from all across the state of Arkansas, which is really, really cool. And it's not just people from across Arkansas who are interested in artificial intelligence per se, and typically you would think about this being computer science oriented. It's also representing very many disciplines as well: biology, computer science, epidemiology, public health. My daughter's actually working on her PhD in epidemiology over at Memphis State University across the river. And when I share with her what we're doing here, she gets super excited about that because she says we're doing really cutting edge stuff. And it leads me to thinking about the new Center for No Boundary Thinking here on our campus, which was approved a couple of Board of Trustees meetings ago. And it's led by Dr. Shushan Wong, who you all know. I don't have to introduce her, but we're super excited about her leadership there and about the efforts for the Center of No Boundary Thinking. What's especially great about that, it is also interdisciplinary. We've got divisions represented from biological systems, advanced data science and learning, and then computational, of course, as well. Shushan is one of the leaders in the country. Arkansas is lucky to have her, and Arkansas State is very proud of her as well. And so I'm excited about this conference. I'm sorry I won't be able to sit in on most of it because we have our board of trustees meeting today, and I'm also doing a presentation right now for the Alumni Association with our new young alumni luncheon today. But I hope I can see a recording of it after, because what you all are doing, what you all are talking about, is really the cutting edge of science and learning and the use and the interface between humans and technology. Suzanne has taught me that computers won't replace human beings, but they will enhance what human beings are able to do, which I think is really cool. So with that being said, I'll pass it back to you, Emily, and congratulations and welcome to Arkansas State virtually, everyone.
Thank you, Dr. Dansis, for the warm welcome. We appreciate you being here and taking time out of your busy schedule. Next, I would like to introduce Dr. Alan Leather, who is our
Emily, you were breaking up, but I am assuming you mentioned my name. Is that right?
Yes, sir.
Okay, great. Well, again, I just want to compliment the Chancellor's comments. I'm Alan Utter, our Provost and Chief Academic Officer here at Arkansas State University. Again, want to welcome you virtually to our campus. I hope somewhere in the future that we can actually have you back on campus, and that day will come. And so hopefully maybe next year we'll be all meeting in person. So again, officially welcome you to the 2020 AI Campus Phase 1 Demonstration Showcase. And as the Chancellor mentioned, we, I've looked at the agenda. There are some terrific demonstration projects that will be presented throughout the day. I want to take just one moment and recognize the institutions that are being represented throughout the presentations. Certainly our folks, our faculty and students here at Arkansas State University, Arkansas Tech University, Arkansas Children's Hospital, the University of Arkansas Fayetteville, University of Arkansas Little Rock, University of Arkansas for Medical Sciences, UAMS, the University of Arkansas Pine Bluff, and the FDA National Center for Toxicological Research. That is a wide range of institutions of higher education representing the state of Arkansas. And again, we are thrilled to have all of you engaged in this project. Let me just say briefly, as a exercise scientist and a former vice provost of research, all of you know this. The road to discovery travels right over top the desk of a scientist. And again, let me say that again. The road to discovery travels over the desk of a scientist, and you know this, it's not a paved road, it's a dirt road. There are rocks and there are potholes, there are hurdles that you have to jump through, but hopefully if you can make it through that journey of traveling down that dirt road, the fruits of your labor are going to pay off in discoveries. And again, it's hard. We understand it as hard work. But again, that is the benefit of higher education, to travel down the dirt road, to engage in questions that folks have yet to answer. Again, when I look at the topics that are going to be discussed today, using AI to interpret scenes for autonomous vehicles, using genomics to genomics harnessing, using AI, biomedical imaging, for example, using AI to contribute to COVID detection and assessment. These are cutting edge topics that would not happen if it weren't for you, the students and our faculty pressing against the current known body of knowledge to extend it. And that is the beauty of an event like this. So with all that said, I want to thank you all for attending. For the students and faculty that are presenting today, I wish you the very best. I actually do have to step out. I have to actually jump on to a COVID meeting of all things. Imagine that. But I'm hoping maybe to join back with you a little bit later. Again, for all of you presenting today, I wish you the very best. And again, welcome to the meeting. Thank you.
Dr. Etter, thank you. Thank
Hey,
you for your time that you've set aside to welcome everyone to Arkansas State University and the AI campus this morning. We truly appreciate you. Next, I'd like to introduce Dr. Tom Chilton, which is the Division Director of Science and Technology at the Arkansas Economic Development Commission. Is Dr. Chilton here?
Can you hear me?
We can hear you now, Dr.
Hello?
Chilton. Thank you.
You went back on mute, Tom.
There you go.
Can you hear me now, Emily?
Yes, I can hear you now.
I'm still not. Can you hear me?
Yes, we can hear you.
Yes, you're on.
Hopefully we've overcome the technical glitch and I'll go ahead and get started. If you can hear me, Emily, raise your hand or something.
Yes, I'm delayed, but
Okay, good,
yes.
good. I apologize for, that's the wonderful thing about technology is it's wonderful when it works and it's dangerous when it doesn't, I guess. Certainly, I'd like to add my voice to those that have welcomed you to this meeting. It's a very important meeting, and it's an important meeting because it's part of a quilt of ideas that are coming together and formulating in Arkansas. As you know, people don't plan, they just fail to plan, and we do have a plan here in this state, and it very well matches up to the activities that are going on throughout the day. One of the things that we're required to do by the National Science Foundation to keep our EBSCOR opportunities fresh and new is to create a science and technology plan for the state. If you've not had the opportunity to review that, you can go onto the Arkansas Economic Development Commission, the website, and you can search on that under the state science and technology plan. And it'll walk you through the various elements that the state is working from in order to enhance the ability for data science and the creation of all the opportunity that comes from that. You know, one of the things that I was thinking about this morning from a virtual standpoint, thank goodness for the technology that allows us to be able to continue business. It would not have been that many years ago, sending people to shelter in place would have meant that they were isolated, and with the exception of the telephone, they wouldn't necessarily have any way of communicating. But with Zoom and other applications that are out there, it's a wonderful thing. But it also removes kind of the human touch element. And one of the things I would encourage you as you think in terms of the artificial intelligence and the capacity and the just wonderful opportunities that get created by that science, that we never lose we never lose the human element. The fact that we need to have that human touch and that technology works for us, not the other way around. One thing that often comes to mind when we're working through our current EBSCOR National Science Foundation grant, which is many of you are working on that with us. It's affectionately known as DART, which is data analytics that are robust and trusted. You know, we all learned in computer science many, many years ago, garbage in, garbage out. And so consequently, we need to have good and trusted information. But in this day and age, it also means that that information needs to pass an ethical test. So one of the things that I would encourage as you talk throughout the day, that every time you think in terms of something that progresses the concept of artificial intelligence, you also match that with the scientific ethic that makes things good or bad. And we obviously see that in our everyday opportunities and working on the net, that things can be used for very good or can be used for very, very bad and nefarious things. So with that, I will turn it back to you, Emily, to continue. We welcome everybody and everybody who's listening from this great state of Arkansas. I'd like you to just remember one thing: you live and you work in a very special place. Thank you and have a good summit today.
Dr. Chilton, thank you. Very wise words. And we truly appreciate your welcome and your insight, especially from that state and economic point of view. Next, I'd like to introduce Dr. Shujin Huang, which is the director of the Center for No Boundary Thinking and professor in the Department of Computer Science at Arkansas State University.
Thank you, Emily. Thank you all joining us today for the showcase. Due to the pandemic, we have the virtual showcase this year. Hopefully, like next year, we will be able to all come to on-site. AI, like a new technology, we work on developing new technologies. The goal is not just for facilitating social distancing. Most important is to bring humans close together. First, I would like to introduce our coach team. For the past 4 months for phase 1 training, our coach team worked weekly with our statewide participants for weekly meeting and also answer question answer on the Slack channel. They all work as volunteers for our Arkansas AI campus. They all here today in real time. And first, I'd like to introduce our chief scientist, Dr. Yuanfang Guan. Yuanfang, would you please say hi, wave a hand, say hi to the group?
Hi, everybody. So thanks a lot for Xiujun to Dr. Huang to involve me into the activities. We are really excited at what we are doing here. Thanks.
Yeah, Yuanfang is associate professor from University of Michigan Medical School. She is very talented, national top AI expert. Next, Dr. Jason Causey. Jason.
Good morning, everybody. Happy to be here and happy to see so many people turning out to watch the showcase.
Dr. Causey is our AI campus coach and coordinator, also associate director of Center for No Boundary Thinking and the division lead for the division of algorithms and computational methodology. Next, Dr. Jake Quaz.
Good morning, everyone. It's nice to see a large crowd today.
Yeah, Jake is also AI campus coach and coordinator, also serves as associate director for Center for No Boundary Thinking and the division lead for the division of advanced data science and learning. Jake also serves as the interim chair for Department of Computer Science. Next, Dr. Wei Dong.
Hello, everyone. I'm glad to see all you guys here. I'm looking forward to work on some of the exciting projects with some of you.
Yeah, Dr. Dong, he is our coach and also our AI campus last year head coach. Next, Dr. Emily Ballas.
Hello, everyone. Thanks for joining to see all the hard work by our AI campus participants.
Yeah, Dr. Ballas is our AI campus coach. also Associate Director of our Center for Newborn Thinking and the Division Lead for the Division of Biological Systems. Next, Lin Rui Kai. Lin Rui here. Lin Rui.
Hi, everyone. Glad to see you.
Yeah, Lin Rui is our AI campus coach. Next, Dr. Kaiman Zeng. Kaiman.
Hello, everyone. It is great pleasure to see you all here. Thank you.
Yeah, Dr. Zeng is our AI campus coach and assistant professor from Arkansas Tech University. Next, Dr. Jonathan Stubblefield.
Hey everybody, it's good to see everybody here. Looking forward to it.
Dr. Stubblefield, he got his MD degree from UMS, is currently working on his PhD with me for AI and biomedical informatics. Yeah, like I said, that all our coach team worked hard with our participants for the phase 1. Today, we will see phase 1 for 6 project demonstration demo, 6 teams demo their work. For next phase, you may know that we are planning to participate the international dream competition for COVID-19. Indeed, actually, our coach team already started the work, and the coach team submitted a test model to the competition. We already get back some interesting result from the test model, and it's all available online on the website. For the real model, our coach team will wait all our AI campus participants to register and join the team to work together for the real model to compete with worldwide teams. And so like, I think next I will turn back to Emily and Jake for us to start the team presentations.
All right. So at this point, we would like to start. We have 6 team demonstrations, and I'll let Dr. Jake Qualls introduce each of these teams and their slides.
Thank you, Emily. So our first presentation today is going to be deep learning detection of COVID-19 through chest x-rays. And this particular team was led by Dr. Jonathan Stubblefield.
All right, so hi, my name is Dakota. I'm a senior in computer science at the University of Arkansas, and I had the opportunity to work on this project again with Dr. Stubblefield. So, okay, just a little bit of background. As I'm sure all of you are aware, we are currently in the midst of a pandemic, and it was on December 31st that a pneumonia of unknown cause was detected in Wuhan, China. And less than a month later, the outbreak was declared a public health emergency. And then on February 11th, we got a name for the actual coronavirus disease as COVID-19. So as of earlier this week, that's when these numbers were taken. There were over 29 million confirmed cases and 926,000 confirmed deaths. So obviously with the widespread nature of this virus, the government is asking for help from researchers, both in terms of treatments and vaccines, as well as diagnostic methods. So the current diagnostic involves inserting a 6-inch swab into the nasal cavity and for 15 seconds for both nostrils. And then there's also an oral version of the swab that is currently in use, but as you can see from this graphic over here to the right, it is very slow and very uncomfortable for the patient. And then once the test is actually completed, the swab is then sent off to a lab for testing. So it's also very inefficient in terms of time usage. There have been current studies done using different forms of biomedical imagery. So CT scans are not usually appropriate for respiratory diseases, and they also result in a higher dosage of radiation than chest x-rays. So for our project, we elected to use chest x-rays. So for this project, we decided to use a neural network to distinguish between the chest x-ray of someone who has COVID-19 and someone who does not. This would effectively eliminate the need for testing kits and would make the testing process much more comfortable for the patient themselves, as well as expedite the entire process. So for our data set, we use the chest x-ray 14 data set from the NIH. It includes 112,000 different images from 30,000 different patients, all of whom were positive for 14 different conditions, as well as there are also some normal chest x-rays in the mix. And these will serve as our positive cases our negative cases, my apologies. Since all of these were released before the pandemic, so there's little to no chance that any of these patients were infected with COVID-19. And then we will mix in positive cases of COVID-19 from another public GitHub repository to solve as our positive cases. And there's also a bit of a transfer learning aspect to this project. And so this graphic just kind of shows the exactly what transfer learning is. So for transfer learning, you have your generic network that has been trained using a generic data set on a generic task. And then you essentially chop off the very end of the network and then retrain it on a more specific task. So for us, what that means is that we took the ChexNet data or ChexNet network, which has been trained on the ChexNet chest x-ray 14 data set, which received an 82.9 ROC score, which is basically a way of determining the performance. You could understand that as a percentage. We removed the classification layer, which while freezing the previous layers in order to retain the model's ability to distinguish between those original 14 classes, added a few convolutional layers in order to give the model some space to really understand how COVID presents in a chest x-ray, along with the binary classification layer, which serves as our positive or negative for COVID-19. Now there is a slight issue with our methodology. So because COVID is still relatively new, there will be a class imbalance. So for example, how that could present in our model, if 10% of our cases are of the minority class, so COVID-19, if the model guesses all samples as the majority class, it will still technically have a 90% accuracy. So for our model, actually, there are 347 COVID samples, and again, over 112,000 non-COVID samples. So these classes are definitely far from being balanced. So there are a few solutions we could implement in order to remedy this. The first of which being to modify the class weights directly. So this would effectively change how the model looks at the classes separately. So it would place more weight on COVID samples over non-COVID samples to avoid it just lumping all of the cases into one class. Or we could modify the testing set composition itself. So as you can see from this graphic here at the bottom, there is downsampling, also known as undersampling, where you reduce the number of majority cases in order to match the number of minority cases. Or there's oversampling, where you can copy the number of minority cases in order to match the majority cases. So downsampling typically improves on your runtime, but it could discard useful information that our model needs to truly understand how COVID will present in a chest x-ray. And then upsampling will typically outperform downsampling, but it's much more prone to overfitting, which basically means that it loses its ability to truly generalize the problem and will become too specific and won't perform as well in practice. What we elected to go with in order to remedy this issue is we went with a balanced data loader. So within a batch size of 32, there'll be 16 COVID and 16 non-COVID samples. And this will operate similar to downsampling in the sense that the non-COVID samples are selected at random and some will be repeated, but all of those non-COVID samples will be used. And so these are our results. So over 172 epochs, we got an accuracy of 0.624, an F1 score of 0.706, precision of 0.587, and a sensitivity of 0.884. And while this may not seem like the best results, simply because of the accuracy being being 0.624, but there was actually another project with chest X-rays called COVIDNet that actually received an F1 score of 0.707 and a precision of 0.547. So we're actually very much on par with that other project. So yeah, I think that's all I have for you today. Thank you for listening. And I think I saw a question in the chat. Dr. Qualls, are we taking questions right now, or do we want to do that at the end?
You can take questions now. It's fine.
Okay.
And what I can do is read it out to you, Dakota. So how we'll facilitate question and answers, please, if anybody has any questions, please do put it in the group chat box. And if you would like to ask it anonymously, you can send it to me privately to Emily Devereaux. And I'll read the questions for everyone just to make sure that everyone will know the question before the speaker provides the answer. But first, for Dakota, from Brian Barnhouse, is it possible to run a mix of upsampling and downsampling data sets? And what are the pros and cons?
So mix of upsampling and downsampling. I guess in theory you could. The only issue, again, that I would be worried about would be overfitting. So by replicating the same sources or your same samples, you run the risk of it learning essentially too much of that sample. And so it will be too specific and it won't be able to truly understand what COVID is, rather than, I don't really know how to word it. So for instance, it could learn if too many of the cases that have been repeated are in a chest X-ray of, say, a male person. It could start identifying what we call COVID cases as a male who has been infected with COVID, is one way that you could think about it. That would be the only issue that I would really see. Yeah, I think that's all I have for.
Okay, thank you, Dakota. The next question is from Fred Pryor. What was the specificity of your model? Distinguishing COVID from pneumonia is difficult.
We did specificity in the beginning of our test, but I don't recall the exact number. Dr. Stubblefield, do you remember what our specificity was?
I don't remember that exact number, but we're using our precision as kind of a surrogate measure for specificity, as that was an easier number for us to get a hold of. Precision, I think, is closely related to the positive predictive value. So I believe you could say that if our test said it was positive, the patient has a 60% chance of having COVID.
Right.
It's definitely more sensitive than it is specific.
Thank you. Dr. Qualls, it looks like we're ready for our second team demonstration.
Yes. Okay, so our second project is the self-driving car project, and they're going to discuss scene segmentation or semantic segmentation in street scenes. So let me show.
Cool. Hello, everyone. Thank you for coming today. So my name is Austin Espejo. I am a senior electrical engineering major from Arkansas Tech University. And so along with my team, Imran, Ashwin, Sadiq, and Marcel, we represent Arkansas Tech University, University of Arkansas Little Rock, and University of Arkansas at Pine Bluff. And so our project is self-driving cars using semantic segmentation in street scenes. And so this is really a crucial component in order to have successful autonomous driving. Okay, so the problem, again, that we are solving is the problem of semantic segmentation, and we are going to be using the full resolution residual network. And so we want pixel level accurate segmentation to create a mask for objects in street scenes.
Austin, you may have to use the controls in the bottom left-hand corner.
Okay. I'm not seeing those. Perfect. Okay. So some problems that we faced in solving this problem was storage for large data sets to train a model accurately. That requires a lot of storage space. So that was one issue that we had. Pixel level segmentation that results in high level accuracy is very difficult. And so we also had the challenge of balancing pooling techniques with convolutional layers. Okay, and so I'm going to go over just some previous works based on literature review that we base kind of our work off. So many models rely on preset weights and biases, and so this strongly prevents the idea of a novel approach in order for training. And so most successful models rely on convolutional neural networks, so this includes many convolutional and pooling layers. And so early models use CNNs to perform pixel-wise classification, and then these pixels are classified in superpixel regions. Fully convolutional neural networks, this allows for a novel pre-train approach, and it also introduces the idea of end-to-end training. And so for SCNNs, there's an idea of skip connections, which allows propagation information from earlier layers to deeper layers. Pooling layers are crucial to increase receptive fields, but output is at a lower resolution. And so work have previously introduced the idea of encoders and decoder pairs to overcome the low resolution problem. And so many architectures rely on the ILS-VRC for pre-training step, but we're going to introduce a novel approach.
Hi, my name is Imran Shokar, and I'm from University of Arkansas Little Rock. Currently, I'm doing a PhD in computer science. So, and I'm also the member of the self-driving car project. And we worked this project under the supervision of Dr. Kaiman Zhang. So for this project, I'm going to talk about the model or architecture that we used for this project. So we used the model, the new model actually, and it's a full resolution residual network. And this full resolution residual network consists of two networks that already been used before, but it's just the merging two networks. And it exhibits actually same superior training, protest, and ResNet, as we know, the convolutional neural network, but it has like a two processing streams. The training program includes the multi-scale context with a pixel level accuracy. One stream carries information at the full image resolution. The other stream follows the pooling operation to obtain the robust feature for the recognition. And this is about these feed-forward networks and residual networks and how these two networks work in this works on the full resolution residual network. Can you go to the next? Yeah, so now I will talk about the feed-forward network. So F and N composed of the linear sequence of the layers, and each layer compute the function F and the output Xn. As you can see from this equation, and here, right here in the F, it actually works as the function, and the Xn is the output here, and Wn is the parameter of the layers. The problem is that it often results in the increased training loss, upper bound training loss, and excessive power in the model. Go to the next. Since we saw the problem with the feed-forward network, so now we added the residual network, and this ResNet convolutional neural network allow network depth that were unattainable previously, and it composed of a sequence of residual unit. So as you can see from this equation, where if xn minus 1 and wn is the residual, which is parameterized, thus instead of computing the output xn directly that we did in the feed-forward network, if only here compute the residual that is added to the input xn minus 1. Go to the next one. And this is the actually full resolution residual unit that is composed of the feed-forward network and the residual network, ResNet. And this architecture, as you can see in the red color and the blue color, and this actually resembles these two networks. Here, Wn are the parameter of the function and A's respectively, and Jn minus 1 is the residual input, and Yn minus 1 is the pulling input. Thank you.
Hello everyone, I'm Ojaswin Jan. I'm a master's student in computer science at University of Arkansas at Little Rock. I'll be taking you through the rest of the slides. So we'll be discussing the dataset. We are using the Cityscape dataset. It's a publicly available dataset. It records images from 50 different cities of city scenes, specifically the streets, and annotates the different images into different classes, 30 to be specific. The recording process includes taking video frames of 1.8 seconds each, and then selecting one of those frames to annotate the images. Next slide, please. So the data set divides the classes into 8 groups, depending on surface, vehicle, whether they're humans, whether it's parts of a construction like building and wall. Different divisions like these help a self-driving car to segment these things and to select path. Next slide, please. Now the training can't be done on the full resolution images because of the size constraints. So they're either done on quarter resolution images or half resolution images. Using full resolution images could be memory intensive and affect performance. Next slide, please. Now the results are like these. So a raw image looks like what you can see on the left. The ground truth, or what would be the ideal outcome of this, would be in the middle. And right is how our results look like using FRRN. Now the data set has 5,000 images. The training set is 2975, validation is 500, and the test set has 1525 images. Now, as you can see on the right side, the images are divided into whether they're pink for sidewalk, red for persons, gray for building. And this is how the entire image is divided and helps a self-driving car navigate. Next slide, please. So the AI campus experience was very great. We got to familiarize ourselves with machine learning and deep learning techniques. In stuff like semantic segmentation, FRN, ResNet. Also, we got to handle a large data set. We got to work on a cloud environment to run our tests. Also, we got to experience working with each other as a team, and that was a great experience. Next slide. I want to thank everyone to listening to our presentation, and we'll be accepting your questions now. Thank you.
Well, that's when you got a question.
All right. Great job, team. Great job. I do have some questions that have come directly to me. And that is one, during Austin's presentation, when the challenges were reviewed, could you perhaps spend a moment on, the rationale of why they are challenges? This level of detail will really help the lay audience that is attending the AI Campus Showcase.
Yes. Sorry, I'm pulling out the slides again just so I can reference. So really storing the large data set. So you really need a large data set in order to train your model, because the better you train your model, the better results you're going to have when you're implementing it. So the large data set is required in order to get an effective model. And so the pixel level segmentation is really difficult as well, just because of the size of the images, and also just the pooling techniques that you're going to need to do and the different layers in order to get that pixel level segmentation. And so the balancing of the pooling techniques of the convolutional layers goes back to the pixel level segmentation, because you're going to be reducing your image size and also some of the crucial details that you need from that image to your output in order to effectively create the mask of the image. I don't know if any of my team has any other points to add to that.
That sounds good to me.
Excellent. Thank you, Austin. That was wonderful. Okay, so Jocelyn,
Yeah,
I'm sorry if I pronounced your name wrong.
It's
Okay, it appears that the image perspective is from a center mounted dash cam. Would it make a difference downstream in your calculations and image processing if the perspective were different, such as the front bumper of the car or the driver's view?
So I think the question would need to be altered, because we are trying to develop this for a self-driving car. So getting images from a driver's view or the bumper of the car might skew how the car perceives its environment. So keeping a neutral position for the camera might actually be the most effective way to look at the surroundings. Companies like Tesla actually have multiple cameras which they use to get images from all the perspectives to map out a proper navigation route and to react to its environment. So I think it's not really a problem of calculations and image processing, but rather whether the image itself is correct for use.
Excellent. Thank you.
And I would like to add more a comment about that question. So if we think about the ascent and the trajectories, and let's say ascent is like the driver itself, and the trajectories are the behavior of the different classes. So in that sense, when you change the direction of the camera, let's say front bumper to the car driver's view, that might make the difference. But in our project, we're not working on that side. So it's just about the image segmentation, and we're trying to level the pixel actually. So yeah, thank you.
Great. Okay, I have another question that came directly to me. What segmentation tool or technique did you use?
So we actually use the image, I think it's the image segmentation. No, actually the semantic segmentation.
Okay. Great. One other question. Has the team considered multi-view geometry, multiple converting images, and how would this contribute to the solution?
No, actually, we did not consider at this point. Maybe in the improvement phase in the future, we can add this to our project.
Can I add to this? So the multiple converting images would just be an extension to this project. Currently, we are only looking at the front perspective, but if we just extend this problem and add a side view to both sides and combine all of these results, then we get to how an actual car will react to its surroundings. So I think it's just an extension of this project and more like a next step. Yeah.
Hopefully in next phase.
Excellent. Great job, team.
Thank you.
Okay. So our next group that will be presenting is the Business Analytics Group, and they will be talking about demand forecasting.
Hello. My name is Jordan Davis, and I'm from Arkansas Tech University, and I'm here to present to you the demand forecasting project. Dr. Qualls, I'm not seeing the controls in the bottom left-hand side. Oh, okay. Well, here we are. I wanted to give you a warm welcome and thank you for taking the time out of your day to be here today, regardless of whether you may have been required or if you're just here out of free time. We really appreciate such a big audience today. So today, our overview for the project is going to be broken into two pieces. We have the purpose and the function. Okay, so our project's purpose was mostly educational. We basically went through, we went and got a solution from a previous competition on Kaggle, kind of similar to the competition that we are wanting to enter in by the means of where it's hosted. So this specific competition was labeled How to Win a Data Science Competition. And we chose the topmost rated response on Kaggle. The task for this solution was provided historical data. We were to forecast sold products in various shops. And from this, a sales model was actually created to answer this question. Okay, so moving on to our function, or the way the project functions, the historical data was housed in CSV files or Excel files. It's a little different from what has been previously presented today, as this is more of numerical data as opposed to image data. And so we used the sales from this historical data for training our model, or for training the model, and the test was set up to use for the prediction. So in this project, there was a focus on a time series with pandas import from Python. So in a couple of ways we used time series was something known as autoregression and moving average. So these two tools provided solid information for predicting near future sales of each store. Okay, I think I am able to control it. I apologize. That might be the issue. There we go. So here you see an example of the data that the model is using for its predictions. This is one example from a singular month. You see here there are multiple data fields from multiple days inside the month of different items. And so this was replicated hundreds of thousands of times, and the model was responsible for taking this in and reading the data appropriately. So here we see a graphical example of the total sales represented by monthly. On the time axis, you see there 0 through 30. So the data had information. The data was from, I think, around January of 2013 through about October of 2015. So that resembles a little over 2.5 years. And so hence why the numbers are up to about 30 months, a little over 30 months. And you see the sales axis is representative of the items sold by the stores. So the first model that we worked with was the autoregressive model. And I think it's important to note that with each of our representations of the models, we use two different graphical representations, and they just had two different parameters just for graphic purposes. So I chose to use the second one of each model. And you'll see here the autocorrelation section. This is for how well the two pieces of information, the time and the sales, correlate with one another. We see that it is regressing as time progresses. But if you point your attention to the probability plot, we see that probability actually increases as time goes on. So that is actually important to note, that the probability of the sales being predicted has a high chance of actually coming true based on the model. So the second model we used was a moving average model. This model, you'll see that is just slightly, it presents the same data as the autoregressive model, but in a slightly different way. The average is taken from the moving average is taken from the actual data itself, as opposed to regressing an average. This average is taken as the average of items sold per month, which is one reason why the correlation is a little more spottier than the previous model. But when you put your attention to the probability again, it's solid as the other probability, which is a good sign for the prediction. And here you see the present forecast. This is the sales per trade monthly resembles the first graph that we saw of the actual sales, but after the black dots that you see on the graph is actually a prediction that the model came up with. So as you can see, the sales were attenuating down, which resembles the historical data and the historical sales that we saw. And like I said, the prediction predicted just a little past the actual time that the data showed us. And looking to the right, you'll see that the sales trend is attenuating down. So in closing, we discussed talking about experimentation with profit, or more experimentation with profit and other time series protocols, and how that may change our data, or how that may change or more accurately portray a forecast of what is to be sold in the near future. Personally, I would like to see maybe what differences in regression we could have if we use something called an autoregressive moving integrated average, as opposed to just the autoregressive model. So that about wraps up our presentation for sales forecasting, and we're open for questions if anyone has any.
Yes, this is Pierre Bichelle from NIEHS, a very good talk. I was just wondering, in terms of the autoregressive model, did you try, or did the Armani include smoothing of the regression, which might give you a better prediction?
As far as I know, I personally did not include any smoothing in the project when I was going through with it. It would probably be very beneficial to do that, actually.
Yeah.
I appreciate that.
I want to give it a try.
Right.
I think exponential models or some other models might work well.
Okay, I'll definitely keep that in mind.
All right. Great job. That was an excellent presentation.
Thank you.
Okay. So our next group that will be presenting is the natural language processing group, and they will talk about exploring COVID-19 in social media, and specifically Twitter.
Okay. Hello, everyone. Thank you all for having me. I'm a senior at the University of Arkansas. I will be presenting the slides for NLP. I have been shadowing the works of Dr. Qualls from Arkansas State University, Catherine Schultz from University of Arkansas for Medical Sciences, and Jennifer Fowler from Arkansas State University. And I'm sorry, I don't know how to. We have used a data set that consists of COVID-19 related tweet IDs. The data collected started January 21st, 2020. This data set only contains the tweet ID for each captured tweet. There were 242,400,994 tweets. And out of those, 157,266,557 were English tweets, approximately 64.88% of collected data. The process data to compile the tweet text and location tweet ID, and we visualize the data with mapping and word clouds in R and Python. Okay, this next slide shows the process of how the data is compiled from the tweet ID. It comes from the COVID-19 tweet IDs from the GitHub, and once it is downloaded, the daily tweet ID files are then converted to the Hydrator. It's a third-party application. Only tweet IDs can be distributed. Hydrator converts tweet IDs back into, hold on just a second, JSON files by downloading original data from Twitter. It is then sent to the pre-processing where removed retweets are extracted elements of interest, and then it's then put into a data set to retain original data files in compressed format. And from then it is converted into text files, location files, and account files. Just a second. Okay, after processing, we have CSV text files for each month starting in January that contains the tweet data of all the tweets in that month. This shows the format. Hold on. Okay, hold on just a second, because I'm seeing different slides. Okay, were there any slides that were prior to this slide?
So I'll jump in here.
Okay.
What this represents is the calculation of the center point for the user location. And this was created by calculating, taking the 4 corner points that was listed within the user location portion of the tweet. And then we calculated the center to identify a potential location for the user. And so once we were able to identify the location for all the users, we looked at the continental US, and on this particular example, you can see, the density for the rate and the amount of tweets that were related to COVID-19 across the US. And at this point in time, this represents May of 2020. And you can see a large area in the Northeast as well as on the Western coast for COVID-19 tweeting. Now, additionally, we looked at the density of tweets coming from the state of Arkansas. And the image on the left represents a single day for the number of tweets that were coming from Arkansas. And then the image on the right represents the number of tweets coming from Arkansas throughout the month of May. So it's not surprising that during this time that the Little Rock area, Central Arkansas, had the most tweets that were related to COVID-19. And, that is contributed probably to the location of UAMS, the tweeting of information from the Arkansas Department of Health. Other significant areas, you can see the Northeast Arkansas area and the Northwest area of Arkansas having a large number of tweets. Now, for understanding this data, we looked at word cloud visualization, and we were able to generate some word clouds as a quick overview of the data. And we can see here from January and then again in May, the wide difference in the number of topics that were being discussed when capturing COVID-19 tweets. So early on, you can see that the most common word that was mentioned was Wuhan and China. And then once we progressed through the pandemic and into May, those topics started to change. You can see that pandemic, quarantine, stay at home became very popular. Now, again, we wanted to look at word cloud visualization by using both R and Python. And in Python, we were able to take advantage of word2vec and utilizing k-means clusters. And for these word clouds that were generated for the month of May, or the month of January, you can actually see a wide range of different topics that were being discussed in the captured tweets. But what was interesting is that there were two clusters that centered around COVID-19, specifically coronavirus hashtags, and then protein inhibitor protease remdesivir was also mentioned throughout January. And here is a word cloud representation for the month of May. And again, we can see a progression of different topics that were discussed on Twitter for this month. Cytokine storm, anticoagulant, heparin, those were particular interests for the COVID-19 tweets, as well as, it was centered around quarantine. So interestingly enough, people were tweeting about quarantines, things that they were doing while being in quarantine. So the next steps for the NLP team, we would like to investigate relationships between epidemiology data and tweet content over time. So a continuation of time series analysis, and then selecting specific COVID-19 related issues for further sentiment analysis. Specifically the issue of reopening schools, how are schools handling the pandemic and reopening, as well as mask mandates. So thank you. So Brian has a question in chat, I believe.
Not necessarily
okay.
a question, just a comment to you.
Okay. Okay. Thank you.
Sure, you're welcome.
Other questions? Okay. So the next group that we'll discuss is the genomics team, and they will be talking about predicting phenotypes through the use of XGBoost deep learning algorithms.
Hello. Hi, hello everyone. So it is really nice to join our AI showcase today. My name is Gwen, and I am an undergraduate student at Arkansas State University. Our genomics team under Dr. Emily Bellis, which include Emilio, Daniel, Kevin, and me. And we really love to participate in the AI campus. And today we would like to show you our progress in this summer. Next slide, please. So our team has conducted 2 projects. The first one is the TCGA pan-cancer project. And as you know, that cancer is a very dangerous disease in which the abnormal cells divide uncontrollably and destroy body tissue. And in this project, we analyze the gene expression data and classify different types of cancer associated with them, as well as the relationship distributed in the data. The second project is the maize genomics project, and we predict the performance of maize from single nucleotide polymorphism markers in a particular environment using XGBoost technique. So far, those are our 2 projects. An overview about our dataset for the first project. It is collected by the pan-cancer project, and it is a part of the RNA sequence pan-can dataset, which is collected by the UCI machine learning repository. And as you can see in the dataset, we have a random extraction of a lot of gene expression of patients diagnosed with different types of tumor, including breast, kidney, colon, lung, and prostate cancer. And we have around 801 patients, and each patient have around 20,000 genes. Next slide, please. So here is just a visualization of our gene expression counts for 100 random samples. And how we analyze it, we use the PCA, which stands for Principal Component Analysis. And now I can show you how it works. So generalizing PCA is a method used to reduce a number of variables in your data, and it helps to extract important one from a large pool of data set. The benefits of using PCA is that first, it helps to reduce the data. We have around like 20,000 genes expression, so we just like condense the information so that we can have an easier task to analyze the data. The second benefit is that it helps to better interpret the data. So from the PCA, we can discover important features of the large data set. And based on that, it reveals a lot of things about the relationships that previously we don't know. So as you can see on the picture is our result with the PCA, and we achieve 94% accuracy. To explain more, if you see from the 0.00, and you see on the principal component 1, which is the most important component, the purple color stands for the PRAD cancer, and also the KIRC tumor has a very significant role in our principal component. Also, if you can see that if from the stem from the 0.00, you can see it can be form a large angle between those tumor. The more or less wider of the angle, you can see the larger correlation. For example, if you can see from the PRAD and also the KIRC, it form a very large angle, which is larger than 90 degrees, which means that it is negatively correlated. Next slide, please. So To evaluate our performance, we use the XGBoost, stand for Extreme Gradient Boost. So this is a form of gradient boosting, and it's a machine learning technique that helps with the regression and classification of questions. So in the XGBoost, new models are created to predict the residual of errors and also added together to make a final prediction. So, and it also helps to minimize the loss when adding new models. And with the XGBoost, we test on this data set and we achieve 99% of precision and also 99% of accuracy. So if you can see, for example, right here in the table, if you have around 61 sample gene expression with the BRCA, it put it the same one like 61 from the beginning, and then at the result, it is still 61 of the BRCA. Or the COAD, we have 25 of the samples, and it's totally classified as the COAD. So I think this is a very good result. And also to take advantage of this XGBoost, we also can evaluate our result by using cross-validation. So in our function of XGBoost, it has the cross-validation methods, and we use a k-fold cross-validation in this project. And as you can see, we have around like 5 k-fold cross-validations. And with each fold, we will test on different data set, different set. So it will produce some mean and standard deviation error. So for example, the first one you see that for the train the data set, we have 0, but with the test data set, it has achieved like around like 0.018 for the mean error and also 1003 for the test error mean. So it's just really, really good result. And so far that's how the first project goes. Move on to the second project, Emilio can explain you.
Yes. So for the second project, we attempted to predict phenotype characteristics of maize plants using genetic or genotype data. So as an overview, we used a data set for CMAs, which is a maize species. And this data set contained genotype data for the 10 chromosomes of maize of approximately 1,500 individuals. Which are parental data for offspring which were planted across several fields in 2017 as part of the Genomes to Fields initiative. So using this data and also an additional data set containing several phenotype characteristics, we attempted to use XGBoost to perform a model to allow us to predict certain characteristics like plant height or grain yield. So the original data was in an H file, which was formatted for a specific software called Tassel. We used this software to convert it to the variant called format, or BCF, simply for an overview of the data set. And then later we converted these to CSV files, which we use in Python for machine learning and XGBoost. Next slide, please. Okay, so just as an overview, this is chromosome 1 of the 10 chromosomes of maize. In the first graph, you can see a lot of lines which represent, each horizontal line represents an individual, and each vertical line represents a position in the chromosome. So all of the white lines are missing data. And in this graph, you can see that we have quite a lot of missing data, and also that the data set is pretty big. So this is, as I said, just of chromosome 1. The blue color represents where the genome, it's in accordance with a reference genome. So that means that it's the same sequence. And the red and the purple represent where we have SNPs or variations across the chromosomes that were genotyped here. In the graph below, you can see more or less how are these variations distributed, and that there are several positions which have more variation than others. And finally, we performed a principal component analysis just as an overview to see how the data was correlated. So you can see that we can see pretty complex interactions in here. Next slide, please. So as I said, we had a lot of missing data. Most of it was due to blank samples, which are simply used for sequencing errors. So as they provide us no information, we simply discarded them. And as for the missing data in each individual, in each position of the chromosome, we calculated the allele frequency for each position, and then we replaced these missing values with that allele frequency. Next slide, please. So as I said, we used this genotype data to predict certain characteristics, but because we had only for the parental maize species, well, we only had DNA sequences for the parental maize species, we had to first predict using this data the sequences for the offspring maize species, which were the ones with which we had phenotype data. So we did this following certain criteria and using the allele frequency. And this allowed us to predict position by position the sequence of each offspring. Next slide, please. So using this data, we selected one field where some of these species were planted. Exactly 174 offspring species were planted in this ARH1 field, which is actually in Arkansas. And we then, after selecting this data, we performed the principal component analysis on just chromosome 1, which you can observe in this graph. And we selected arbitrarily just 2 principal components for training a preliminary model in XGBoost. So using these principal components as the input variables, which were our genetic data, and the plant height as the output variable, we predicted, we trained a machine learning model using XGBoost. Next slide, please. So in this first query, you can see some of the parameters we used for the XGBoost model. And using these parameters and the data that I just mentioned, and evaluating the mean absolute error for accuracy, we obtained for this preliminary model mean absolute error of 14.76. And as a baseline, we compared it with predicting just using the mean, which showed a mean absolute error of 16.73. So we actually improved the prediction using the XGBoost model. So as I said, we just arbitrarily chose the field and the principal components that we used. So in our further development of the model, we can actually use a larger amount of principal components as explained here in the variance plot. So for example, we could use 70 principal components in order to retain at least 80% of the variation or the important data in our genotype dataset. Other things we can do to further improve this model is the incorporation of data from the other 29 planting fields that there are. As I said, we just used one of these fields for this model. And we can also implement other feature reduction other than the principal component analysis that I just mentioned. We can also filter the rare SNPs in the genotype data and focus more on the ones that actually are present along most of the population. And finally, we can also identify the SNPs resulting from environmental factors, which we can do correlating environmental data with each of the fields where the seeds were planted. And that's all I have for you. Any questions?
Very good, Emilio. Thank you. So do we have any questions for the genomics group?
One just came through. How did you optimize your XGBoost models?
So,
you can go.
All right. So for example, for optimizing the XGBoost models, I used k-fold cross-validation also, testing each of the parameters that were shown in the last square of the slides. And by doing repeatedly k-fold cross-validation in these parameters, I was able to optimize most of them.
So mostly that if you want to optimize the XGBoost, so the approach that we used is that we cross-validate the data set. So instead of you just divide the data set into two single train and test data set, use the cross validation, you can just so like how to say you just divide, you just choose a k number. So in this project, we choose a number 5, which means that it will analyze the train data set into 5 times. So from one single data set, you can just like mix it up and then do it 5 times, and we can see different results, and you can see in the tables. And that's how far it goes. We also use, we think of the normalizing the data, and we hope that by doing that, it will help to produce better results. And until now, we're still looking into it. So that so far that's how our project goes.
Okay, any other questions? So
one more.
yeah.
Here's one. While classified as a diploid, maize AHS abnormal chromosome numbers such as tetraploidy, did you model account for this?
So the data set we had for the maize data only contained data that was classified as diploid. So the rest positions that may have had several, that may have shown tetraploidy or something were not present in our data set. So we simply used diploid data.
Thank you.
Okay. Then the final presentation that we will have is from the biomedical imaging group, and they will talk about the deep lesion data set.
Hello, this is Chenghui Li. I'm a faculty at University of Arkansas for Medical Sciences. I would like to thank Dr. Huang for having me in the AI Campus project. And also Dr. Kauzi is the leading faculty for this project. He was really very helpful to help me understand and even setting up the Python. And Dr. Quills was overseeing the whole AI campus. And also I presented my research project proposal for using Twitter data. So I worked with him on the Twitter project, although not the ones that they are presenting, but kind of in the parallel process. So I'm very thankful for all the faculty's help. I want to first saying that among all the participants, I'm probably the least experienced with computer science and AI techniques. So what I'm going to presenting today would be really very basic things that I've learned through the summer, and especially the bioimaging part. That's not just the AI, but just understanding how the computer seeing those images. That's something very new for me. I realize I'm the only one representing UMS, and I have to say I'm not a good representation of the UMS today. The reason I'm interested in this bioimage project is that I'm working on a project looking at cancer patients, and we want to particularly looking at the radiation therapy and how that affects the heart. There are not a lot of detailed information on the therapy itself. So what we want to see is that how the radiation would affect the substructure of the heart. So for that project, we actually wanted have collaborators that are radiation oncologists to try to contour the heart to be able to identify those substructure. So this project would help me understand, how the CT, all these imaging data, how the computer read those data, recognize those structure. I don't think right now I can write a program and be able to identify those substructure automatically, but I think the knowledge I gained from this project and hopefully in the next phase of the project would help my understanding and help with researching that project. So, just a little bit of background of the lesion detection. So we're using CT scans to try to identify the lesion. The detection of the lesion or other abnormality in medical image is a valuable part of the diagnosis. And in the past, most of the existing data may be targeting one particular lesion type. The right image you can see is a CT scan, and there's a system called ResIST, which is a standard imaging tool that can identify the tumor, the size of it. This is used to monitor the progression of the disease in terms of the changes in size on the image. So the data set we are using is a subset of deep lesion data set, which contains 32,000 over 32,000 computed CT slices from over 4,000 unique patients. The slides will, there are usually 1 to 3 lesions in each image with accompanying bordering boxes and size measures. So the total lesion actually more than the number of CT slides. So there are, so for this exercise, we actually used just a small subset of the database simply because the original database, because its imaging files is huge. So the sample data that we're using included about 1,300 CT slides. So they contains the key slice that has a lesion, and also other slices that included the region 30 millimeters above and below the key slide. The details about the data set is available at KGO, and the programming, the programs that we use is also from that website. All right, so just a little bit of information about the data. So these slides are annotated, and they include a patient age and gender. For each patient, that could be 1 to 26 studies of the patient image. And then they include a key slides, and also, as I said, also bordering slides. If the tumor actually huge, they will include additional slides just to make sure that they cover the whole area. In the data, it also has the dimension information. So I think that that's what's used in the program to be able to identify the ground truth for the training, to be able to see how to develop the AI algorithm to better identify those lesions. The data also included 8 different types of tumors, bone, it's all here. Just for the interest of time. For the program that we used, these are the packages that we specifically used to run those programs. So just to show you an example of the image, so you can see, the program actually using those data on the location of the tumor to try to develop a kind of square, a box area to mark where the location of the tumor is. So this is an overview of all the CT slides in the data. So the algorithm that I used to the AI technique to use to be able to kind of identify the lesion on the image is called UNet. So UNet works by learning a latent representation of the image and then reconstructing a new image of the same size that focus on the task. So as you can see, this is from the original paper of the author who developed this algorithm. As you can see, it has this U shape. So basically it has two processes. One process is through the decoder process. So it's the convolution process that through the different filter to try to focus more on the specific tumor. And then I have to say my understanding is still very limited. So but in the process, you kind of down sampling the imaging data. And then the other process is the recoding process called transpose convolution, where you kind of reconstruct the new image, but with the emphasize the tumor area, but also the surrounding area. So, so go back to the original size of the image. So for me to, it is it's taking me some time to understand the algorithm. I have to say I have not fully understand it. But just, for those who are not in computer science and not familiar with this, the next few slides may be of a little help. At least it was helpful for me. So when you identifying a image, so the first maybe the simple step may be to say if there's only one image, like there's only one item on the image that you want to identify. The basic task is to say there's a cat there, and then the next one is you want to not only see there's a cat, but you also want to see the general location of that cat. And then for image that maybe have multiple projects that you want to identify, and then you want to distinguish the different project. And then you can see from a previous slides, it was just a general box marking those objects. And then you want to further be able to kind of really showing the general shape of it, and also distinguish the different type of project. And then further is that, even for human, there are different humans, and you also want to be able to differentiate the different individuals. And for tumors, there may be several tumors bothering each other, and your algorithm want to be able to distinguish the different tumor on that image. And that's just an illustration of, from the object detection to the being able to identify the incidences. So this is, I think the CAR project has similar ideas, but this for biomedical imaging is also from an input image. From an input image, you want to be able to specifically identify the different type of objects and be able to mark the exact location of that on the image. So what is a convolution operation? I think for most of you, this is really, really very basic, but it's just my basic understanding of it. So essentially you have an input image, and then you go through different layers, try to focus on the things you're interested in. And then that, after the several layers, you come up with the output image. So this is a kind of a illustration of the process. So you have this input image that has 7 by 7, and there's an out layer has some padding, and then there are 3 channels, and then there are 2 filters to go through that, and then after that you have the output value. So when I look at the convolution, essentially you're performing, you're kind of identify which area you want to focus through the numbers you assign to that. So for instance, maybe -1, you want to really not focusing on that, discount that, and where 1, you want to really show, emphasize that image. That's just my basic understanding. It could be wrong, but so that's the convolution part. And then, so for the UNet, it uses a process that try to emphasize the feature that they want to emphasize on. So they were using the maximum pooling operation. So this is a simple illustration of that. So suppose you have this initial input information, and through the maximum pooling, you want to identify through each of the filter the maximum value. So for this one, for this square, the max is 6, and for this, that's 8. So through this simple kind of 1-layer filter, you come up with this final feature map that only include those that have the maximum value. And then the transpose convolution is to reverse that process. Not exactly reversing, but it is the opposite process of normal convolution to be able to restore the original image with not restore the original image, but just to an image that will be high resolution, but still include the ones that are identified through the process. So this kind of illustrate the process of going through convolution side of it, and then transpose convolution side. So as you can see, going from here to here, it is downsampling, emphasizing the part that you want to emphasize, and then this is restoring to the original size. So this algorithm that we use is not exactly the original UNet. It's a simpler application of that. And so I think they divided the slides into a training set and testing set in a 7:3 ratio. And so this is the image. I think these two are the, it seems to be showing at least this one was the same image, but more focused on this area with the tumor. And I think this side was the ones that marked the tumor location. So as you can see, kind of to me, I think this is kind of making the parts that are not important to be less, kind of only emphasizing the part that has a tumor. Anyway, so so after they go through the process, and then they have to evaluate how well the algorithm performs. So they look at the loss function, and they look at the DIC coefficient, and that's essentially measuring the overlap between the predicted and targeted masking area. And then positive accuracy, assuming that's the percentage of a true positive. And then the binary accuracy, I didn't dig out the exact formula, but I'm assuming that it included both positive, true positive and true negative predictions. So this is the evaluation part that shows how well the training set perform, and then the validation set, using applying the algorithm performed. As you can see, once you apply to the validation, it's not as good as the training set performs. So in the next slides, you will see the original image, and then you see the ground truth, and you see the prediction. So graphically, this helped me understand what that is. So ground truth, it's ideally it would be exactly marking where the image should be, but your prediction may not be that good, and as you can see, it kind of missed the target a little bit. So this is that image, and I think then sorry, and then the ground truths you can see is just a couple blocks as we have talked about before. You know, that's the information in the data that is you know, you have this information to be able to construct the box that mark the tumor, and that's the prediction. It seems like it, you know, it's quite a large, wide area compared with the ground truth, so it's not very good prediction. So the limitation, the data does have limitation, and will have a 2D diameter measures and a bounding box of lesion, so the information is limited. That's part of that's contributing to the algorithm doesn't not work very well. That's an important component to that. Okay, so I think that's my presentation. I know this is very, very basic. I'm still learning, and I hope. I mean, yeah. So that's the end of my presentation.
Yeah, Chen Hui, she is an associate professor from UMS College of Public Health. Besides this image project, she also worked on the natural language processing project, and she recently got a grant from UMS internal computation. That's great. Yeah.
I just one correction. I'm with College of Pharmacy. These are the references that I used to prepare for this presentation.
Very good job, everyone. Thank you.
Great job. All right. So let's go ahead and go to the short address to try to keep the meeting on schedule to get you out of here by 1. Dr. Carl Walker is going to be representing the Arkansas AI Campus Board, and he is not only a board member of the Arkansas AI Campus, but he's also chair of the Department of Mathematics and Computer Science at UAPB.
Thank you, Emily. I appreciate that. Also, I want to say excellent presentations from everyone. Excellent work. I think there were some outstanding topics, and I want to thank all the coaches and the teams for their hard work on all of the various topics and projects. I also want to thank Dr. Wong for involving me in the AI campus. I appreciate the opportunity to serve as a board member of the Arkansas AI campus. This is an exciting opportunity for me. But more importantly, I'm excited that some of my students at UAPB get a chance to take part in this unique program. Also, they get a chance to work with some of the top AI experts in the country, even around here in Arkansas. And they also get an opportunity to work with some other brilliant students at other Arkansas institutions, 8 different Arkansas institutions here in the state. And I think that's excellent for our students at UAPB. UAPB is a small HBCU in Southeast Arkansas. And not only that, I'm also excited that there's a possibility of expanding the Arkansas AI campus to be a nationwide AI campus, you know. And so that would also allow an opportunity for other HBCUs like ourselves to get involved. And so I was excited about an opportunity to invite several HBCUs from around the country, other computer science departments, to come and watch the showcase and see what excellent work our students are doing here in Arkansas, and to see if we can replicate that around the entire country. And again, don't have much to say, just excited to be a part, and thanks for the opportunity.
Thank you, Dr. Walker. Next, I'd like to introduce Dr. Sarah Naring. She is the director of the Internal Medicine Residency Program at St. Bernard's Medical Center, and also is a co-director with Dr. Wong for the Translational Research Lab.
Thank you all for inviting me. I'd like to say I think everyone did a really great job today. I'm just blown away by the innovation and the great ideas that are coming out of these conferences. we are currently looking at, of course, COVID with a lot of things. And one of the things we noticed was that COVID-19, there are a lot of people who present with respiratory symptoms, become very sick and die, but there's also a good number of people that are presenting with less severe symptoms and often become the ones who are the spreaders because of the lack of the traditional symptoms. So we're looking at developing a data set, and from that, a predictive algorithm that helps determine who has COVID that may have a milder presentation. For example, one thing I hear pretty commonly is, I just thought I had allergies. So we thought if we could develop a highly sensitive and specific predictive clinical model that would help identify people with minimal symptoms, we might be able to slow the spread even more. So I'm looking forward to working with Dr. Wong on that project. We have worked together before on respiratory related issues, and the machine-based learning that we've used for those projects has been greatly helpful. And so I feel like this is another area in which this will become very helpful. And again, thanks for having me here. And I'm really excited about all the things I heard about today. And I'm looking forward to hearing more come out of you.
Thank you, Dr. Neri. We appreciate you. Next up, I'd like to introduce Dr. Jackson Cawthorn. He is the director of the Arkansas High Performance Computing Center.
There we are. Thank you, Emily. And I wanted to add my congratulations to all of the projects that were presented here today. There's a lot of interesting work going on, and I'm looking forward to seeing more. This is my first AI conference, and so Sujin, thank you for inviting me. And I just wanted to say a couple of things. Tom Chilton mentioned early on the data analytics at a robust and trusted EPSCoR grant that started in July. That is a complex grant, but it's centered on data science work in the state. And there are some very important questions that we're addressing in pure data science research. But for this group, I wanted to point out a couple of other opportunities and work that are associated with that. So one is that, as many of you have pointed out in your presentations, this is a compute and memory and storage intensive research. And you need access to large computing resources to make that happen. One of the goals of DART is to make the resources that are already in Arkansas and new ones that are added by the grant available to a much wider group of people. And so we've begun that. Sujin and some of her students have accounts at the Arkansas High Performance Computing Center. We're working very closely with Fred Pryor at the UAMS High Performance Computing Center to combine those resources in a virtual environment and to provide access to any student and faculty in Arkansas who needs it, and to large memory compute nodes, to large storage arrays in the petabyte range, enough capacity that will get you started. It may not answer all of your questions. You may need to go to larger resources that NSF and NIH provide, but it does get you started on that. And so there'll be a lot more information coming out about that in the coming weeks and months. So I look forward to working with this group as part of that program. And Sujin's one of the leads on that, as I am and Dr. Pryor at UAMS. And if you have any questions about that, please get in contact with us and we'll start to answer those as we can. And a lot more information will start coming out as we ramp up the marketing of the project. So thank you again for all the work.
Dr. Cawthorn, thank you. Thank you for those kind words and for everything that partners have done for the Arkansas AI campus. This has made it to where it is today, and we cannot express our thanks enough. Saying that, next up, I would like to go ahead and introduce Brian Barnhouse. He is the Chief Operating Officer for the Arkansas Research Alliance. And the Arkansas Research Alliance is generously sponsoring the awards for the Arkansas AI Campus Phase 1 Showcase today. And Brian, please take the house. Everybody's counting on, everybody's listening.
All right, here comes the cash, I guess. Thank you, Emily. Thank you, everyone. I'm also proud to serve on the AI Campus Board, and I appreciate the invitation for ARA to be able to sponsor these awards. You know what I love about this whole project, the whole idea behind no boundary thinking is that the teams are cross campus, they're cross disciplinary. You guys are tackling a variety of fields in which to apply these tools and this thinking. And it just really speaks to the nature of what is at the core of the Arkansas Research Alliance. And for those who don't know, ARA is a 501c3 public-private partnership set up 12 years ago, mainly to help with the recruitment, retention, and recognition of 5-star scientific talent to Arkansas universities across the state and to NCTR. And you know, we've been living into that very well. Sue Shen is one of our most recent fellows over the last couple of years, and she joins a cohort of over 25 scientists across these 6 institutions that make up the ARA Academy of Scholars and Fellows. And it's with that in mind that I want to offer everybody, before we get to the awards, I want to offer everybody another opportunity to jump on a webinar next Wednesday at 11 a.m. It's September 23rd, 11 a.m. I'm going to put the
Cannot hear you, Brian.
Brian, you muted.
Sorry, I muted myself when I thought I was putting something in chat. So here we go. So I want to offer everybody the opportunity to jump on a webinar next week, something called Project Scope. We're asking each of our Academy members to present and host a very scientific deep dive into their world once a month over the next several months. So this is a way to register in the chat link and really reach out and understand that there are opportunities for students, grad students, postdocs, and researchers to have a very successful, rewarding career here in Arkansas involved in some cutting edge, frontier busting research. So with that in mind, let's jump into the awards. First, we have 2 categories of awards. First up is the Distinguished Performance Awards. And as I understand it, the certificates will be coming in the mail via Emily Devereaux, and the actual check will, since I don't have a big one to hold up, it'll have to just come via ARA. We'll make sure that hits your inboxes soon. So the Distinguished Performance Award recognizes participants who demonstrated creativity, initiative, and perseverance with learning how to incorporate AI into their respective field of study. So from Arkansas State University, we'd like to recognize Quinn Ho and Jennifer Fowler. So congratulations. There's my clapping for you.
Thank you, guys.
Thank you so much.
Wow.
Next up, from Arkansas Tech University, we have Jordan Davis and Austin Espejo. Congratulations. Well done.
Thank you.
You're welcome. This is very surreal. Now I know what it's like to.
Thank you, sir.
You bet. From the University of Arkansas, Distinguished Performance Award goes to somebody I quizzed on this webinar, Dakota Dale. Well done, Dakota.
Thank you.
Good thinking on your feet out there. Next up, from the University of Arkansas, Pine Bluff, Marcel Noyukwa. Way to go, Marcel. Nice job.
Oh,
and next in line for the Distinguished Performance Award from the University of Arkansas at Little Rock, Kevin Chappelle and Emron Sarkar. Good job.
Thank you very much.
You're welcome. Welcome. And last but not least, for the Distinguished Performance Award, we have from the University of Arkansas for Medical Sciences, Catherine Schultz and Cheng-Wei Lee. Congratulations, Kat and Cheng-Wei. Excellent. And for the Pistas de Resistance, we also have a final award on a Rising Researcher Award, and we're happy to recognize this award for the participant who demonstrated exemplary performance and contribution to the overall success of this Phase 1 project. And from Arkansas State University, Emilio Soriano Chavez. Congratulations.
Thank you.
You're welcome. That was a lot of fun. Thank you for inviting ARA to do that. Appreciate it.
Brian, thank you very much.
Yes.
We truly appreciate ARA, and thank you to what you have brought to the AI campus and for serving on the board. Thank you for your service. Right, to close out this event today, Dr. Abhijit Bhattacharya and Dr. Thomas Risch. Dr. Bhattacharya is the Dean of the College of Engineering and Computer Science at Arkansas State, and Dr. Thomas Risch is the Vice Provost for Research and Technology Transfer at Arkansas State University.
Thank you, Emily. It was really exciting for me to be part of this event, fabulous event, not only because it's held on a Friday morning and we go into the weekend, right? But it was also exciting because I could not attend this event a year back when it was held in Little Rock. I wanted to, but because of other commitments, I could not make it. Now, what strikes me every time, I mean, we all know this, but what strikes me every time is the all-pervasive nature of AI. And that was captured by the 6 different presentations, ranging from self-driving cars to COVID. And if you look at what's happening in the national level, and I looked it up this morning, many of you know that AI, artificial intelligence, is a national priority, and it's been institutionalized by the president in his executive order 1.5 years back, announcing and creating the American AI Initiative. And I think it's fair to say that what all of you are doing, the students, coaches, Dr. Wong and her colleagues in the Center for No Boundary Thinking, Dr. Qualls, Dr. Causey, and Dr. Bellis, is that you're not riding the wave of AI, but you're actually creating the wave of AI in our natural state. And it's splashing all over the state itself. So, and more of this is going to come down the road. So I really appreciate the opportunity to be part of this fabulous event. Wonderful job done by the students and the coaches. Look forward to more of the same, going forward. Thank you all very much. Dr. Risch?
I'd like to thank you all for attending today, and would like to thank the board of the AI campus, the ARA, for their sponsorship. And I need to add my congratulations to all the presenters and participants, and also the coaches. The presenters and the participants made the coaches very proud today. I can assure you it was fantastically done. So some of the future of our AI campus, in the short term, we look forward to the phase 2, the international competition. The last round of our AI campus, the phase 2 did very, very well in their international competition, and we look forward to hearing and seeing at the phase 2 presentation in a few months. Also, as Dr. Walker mentioned, we plan on taking the AI campus from Arkansas to the nation, and we're actively pursuing partners and grant opportunities to do so. And we feel very confident that that's something that will be on the near horizon for us. And finally, I'd like to address the Center for No Boundary Thinking. The range of presentations and the lack of boundaries between the disciplines was well on display today. Now, here in Arkansas, we're the home to the first Center of No Boundaries Thinking, but I assure you that we won't be the last. So I have it on good authority that there's other universities looking at this idea, and we can take pride in the fact that this group here was essential in creating this whole concept of AI. So again, congratulations for everyone, and for all of you that came today. Thank you so much for tuning in. We appreciate it.
Thank you all.
Thank you. Are we done now, or?
We are done.
All right.
Have
Well,
a wonderful
congrats.
weekend.
Thank you. Emily, thank you. And
Thank you, everyone.
Dr.
Great
Risch.
job.
Thank you. Yeah.